Thank you very much for inviting me. You might wonder, what's a plastic surgeon doing here amongst so many eminent orthopedic surgeons? But rather than view me as an interloper, perhaps it's more appropriate to think of me in a more positive sense as someone that Dave is prepared to consider, because I think he's a holistic orthopedic surgeon. He looks at tissues beyond the bone. So I've been asked to talk about the biology of fracture healing, and I'd like to come at it from a different viewpoint in terms of the small molecules rather than the cells. So this is part of my clinical practice, treating high-energy fractures, and they're characterized by an absence of cells which may help the bone heal in terms of periosteal stripping. Often the orthopedic surgeon may remount the medulla and then wash away all of the endogenous cells and proteins. So the first questions we asked was, did it matter with which tissue you cover this bone? And in general, the clinical series are unhelpful because they're retrospective reviews. So if you look at the experimental work, so this is a group from Norway. They put some PTFE in between bone and muscle in a rat femoral fracture model. And they found that if you put the PTFE in early, then you delayed fracture healing. But if you didn't insert it until 2 weeks after the fracture, it had no effect. This group put a nitrocellulose membrane in with pores up to 50 kilodaltons, and they found delayed fracture healing. And the same Norwegian group, if you excise the muscle lying next to the fracture, you could delay healing. But if you then replaced it with muscle from the same animal, but from a nearby site, that restored it. But crushing the muscle didn't have any effect. And this group from Australia has reviewed various preclinical and clinical data to support this concept. Until recently, the group that's really looked at this in a systematic way was Emil Schemitz. And what he did was, in a canine model, took out a 5-centimeter segment of tibia, then replaced it, plated it, and in 50% of the group, he covered the anterior 1/3 with tibialis anterior muscle. And in the other group, he simply closed the skin, but he'd excised the fascia beneath it. So he had a skin group and he had a muscle group. And using various healing parameters, he showed that the muscle group healed better than the skin group. The problem in all of this is, of course, in both groups, 2/3 is covered by muscle around the back. And furthermore, there are a limited number of molecular studies you can do in a canine model. So what we did was develop a murine model to emulate the human situation, where you've got periosteal stripping, so that's the knee and that's the ankle. The soft tissues have been stripped circumferentially. That's the fracture site. There's an intramedullary nail following reaming. It's not locked, of course. And you can't see it with the naked eye, but if you look at it with micro-CT, there's comminution. So it's not too dissimilar to the clinical model. And what we did was, in half of the animals, we put some PTFE in to exclude the muscle at the back, and in the other half, just exclude the skin on the front. And we compared healing. And here it is with micro-CT, but perhaps best shown by bend testing. So this group, there was no PTFE, so that's the control group. And you can see that the muscle group is much better, 3-fold greater breaking strength at 4 weeks post-fracture compared to the fasciocutaneous group. So there's something about muscle which seems to aid fracture healing. We looked at the vascularity, and in both groups, the vascular density in the tissues increases post-fracture. But interestingly, more in the fasciocutaneous group. Now, you can't measure blood flow in a mouse very easily, so we use this as a surrogate measure. But I don't think it's the blood flow in the tissues. When we took tissue from adjacent to the fracture site, so this is from the muscle at 3 days and then cultured for 7 days, stained with this blue stain, or you can continue the culture for 28 days and then you get bone nodule formation. We see lots of bone-forming cells in the muscle, but very few in the skin. And if you do all of the soft tissue dissection but don't break the bone, you don't see any response. And obviously in the controls, you don't see anything. So it seems that muscle's got lots of cells which like making bone. And this has been previously shown, so you can take muscle-derived stromal cells or stem cells, and if you put them in osteogenic medium in vitro, they make bone nodules, and if you put them in adipogenic medium, they make fat, and in chondrogenic medium, they'll make cartilage. And if you look at the cell surface markers, these cells are essentially stem-like, they're not endothelial, and they're not hemopoietic. We took some of these cells from mice that are EGFP positive, so all of their cells glow in the dark when excited by a particular spectrum of light. And we injected those in the posterior calf of a normal mouse, and if you look at it at day 14, so there's the fracture site, there's the skin, that's the bone, You can see the healing callus here, and under fluorescent light you can see that some of these cells have migrated to the fracture site, suggesting that they have some involvement at least in the fracture healing process. Go back to the human then. So sometimes my orthopedic colleagues discard pieces of bone that are non-viable from these patients with fractures. We put them in some serum-free medium, we filter-sterilized it, and put that filter-sterilized medium onto human muscle-derived stromal cells or cells from other tissues such as bone marrow, fat, or skin, all stem cells. And we found these interesting data. So muscle-derived and bone marrow-derived stem cells are very keen to make bone, whereas fat and skin-derived stem cells are not. And here it is the same data shown with bone nodule formation. But actually what much more interesting than where these cells come from is what is it in this medium that's leading to this differentiation. BMPs are, of course, known to be important, and if you add recombinant human BMPs to muscle-derived stromal cells, you get an osteogenic response. But when we added antibodies, neutralizing antibodies to the BMPs, to the supernatant, in fact, there was no inhibition. So it doesn't suggest that BMPs are not important. What it shows is that there is something else. We then made quite an interesting observation. In some patients, you can't reconstruct them, and they need an amputation. So we did the experiment as I've just shown, but in the patients undergoing amputation, distant from the fracture site, we atraumatically cut some of the bone slices, a lot like cucumber slices, and did the same experiment. We found this, that the fracture patients were variably active in terms of bone formation or in terms of attracting the cells across a transwall membrane. But the surgically cut ones were never active. And we wondered, is it the inflammation at the fracture site which is driving this process? We measured the common pro-inflammatory cytokines, TNF and IL-1. They were below the threshold of detection of the Luminex array. IL-6 is up, perhaps slightly higher in the fracture group than the non-fracture group, but really no clear pattern. So we did the same experiment as shown with the BMPs. We added neutralizing antibodies here to TNF. And you see this dose-dependent decrease in osteogenic activity. You see the same effect with IL-6 and a slight trend down with IL-1, but not significantly different from the isotype antibody control. When you do the opposite experiment, when you add recombinant human TNF to human muscle-derived stromal cells, you actually see an osteogenic response, but at very low doses, so optimal at about 1 nanogram per ml. Now, TNF is known to be bad for bone healing, so that's what everyone thinks about in terms of rheumatoid arthritis and where it is now standard therapy. But that's at much higher doses, and you can see that it falls below the baseline. IL-6 has a similar positive effect, but at much higher levels, and you can see the effects here in terms of bone nodule formation for both of those. IL-1 is interesting. It leads to an osteogenic effect in terms of alkaline phosphatase production, but doesn't go on through to completion. We did a similar experiment in terms of did these cytokines have an effect on cell migration. You can see here with anti-TNF inhibiting cell migration, anti-IL-6, so this has been well described before, nothing with anti-IL-1. But if you add recombinant TNF, you see a migratory response. It acts as a chemokine, but now at 1 picogram per ml. And at the optimal osteogenic dose, it actually stabilizes the migration. And you go higher, it repels them. So you can imagine the scenario where you have an injury and distant from the injury site, you have very low concentrations of TNF, and the cells may migrate up the concentration gradient and then undergo osteogenic differentiation. So that's what the data show currently. We measured the amount of cytokine release into the bloodstream, and we found very low levels of most. The TNF was below the threshold of detection. You don't get enough tissue in a mouse model to measure the cytokines directly at the fracture site. But we instead looked at messenger RNA expression. You can see tiny amounts of TNF being expressed. So there isn't a lot of it around. So we wondered, what if you added a bit more? Would that help? So this is the untreated mouse at day 14, and you can see the fracture site. That's the healing callus, that's the chondrogenic bit, and here's bone formation. It's always a little bit faster on the muscle side. And if you give TNF only within the first 24 hours of injury, this is at day 14, you can see this bridging, and it's remodeled. And there's the data with micro-CT at day 28. So it seems that stimulating with a particular cytokine very early on in the process, you can actually take the process through faster, but through to completion. So you don't get a super bone or a super mouse. You just seem to get that. You seem to get the same physiological healing, but faster. The in vitro data I showed you suggests that the therapeutic window might be quite narrow, but in vivo, in this model at least, there seems to be at a 100-fold window of opportunity. If you add the anti-inflammatory cytokine recombinant IL-10, you get an inhibition. So it suggests that that initial inflammatory response is very important. I don't need to remind you of the importance of fragility fracture. So we wanted to know, does the same paradigm pertain to osteoporotic bone? And if you take the ovaries out of female mice, they become osteoporotic within about 4 to 6 weeks. So we repeated the same experiment with those, and you can see that if you add again TNF within the first 24 hours of the fracture, you get accelerated healing at day 14, but at day 28, they become fairly equivalent. And here are the micro-CT reconstructions at day 14. So this is with the control group with PBS and the TNF-treated group. So in summary then, I think the soft tissue envelope in contact with the muscle in contact with the bone can significantly affect the fracture healing. And I think we need to start considering the biology of the local environment in terms of the cytokines, because these can be manipulated, I think, to perhaps influence the endogenous healing processes and the stem cells which are undoubtedly involved. Thank you.